Investigating the Effects of Neurexin-1 Deletion on Neuronal Connectivity by Ethan Howell. Autism spectrum disorders, also known as ASDs, affect communication and often present as repetitive behaviors in children and adults. As the image on the right shows, it can also contribute to a number of other comorbidities, including depression, anxiety, and ADHD. Severe syndromic cases of the disorder, such as Pitt-Hopkins syndrome, can dramatically impact the quality of life for those who suffer from the disease. More often, autism presents in a non-syndromic form, which can be highly variable. The exact genetic background of ASD is still being teased apart, and a better understanding of the myriad of disorders would help to inform clinical approaches. The Neurexin-1 gene has been identified as a possible contributor to an ASD phenotype. Along with other identified synaptopathies, such as mutations in the shank and neuroligin genes, Neurexin-1 mutations appear at a higher rate in populations of patients with autism. Neurexin-1 codes for neurexins, which are important cell adhesion molecules in the presynapse. They play many different roles through helping to regulate the release of neurotransmitters and facilitating communication with postsynaptic molecules. For this reason, mutations in Neurexin-1 are predicted to be severely impactful for the cell's ability to communicate across a synapse, and this loss of function could contribute to the phenotype seen in many autism spectrum disorders. To further complicate Neurexin-1's role, it is also found to be spliced at high levels across its three isoforms, resulting in thousands of potential splice variants. The human brain has two main categories of signals sent between neurons: excitatory and inhibitory. Excitatory signals attempt to trigger an action potential within a neuron, while inhibitory signals suppress such activity. Different neurons in the brain work to trigger either excitatory or inhibitory signals, and an imbalance with this ratio can greatly impact higher functioning. As a result, the excitatory-inhibitory balance is highly regulated, and it's theorized that autism spectrum disorder is a result of this balance being tipped in favor of excitatory behavior. By encouraging the release of greater excitatory signaling, a Neurexin-1 loss of function may contribute to the presence of autism spectrum disorder in affected patients. To analyze the effects of a Neurexin-1 knockout on synaptic communication, the cell lines of patients with ASD were used. 3 patients with ASD served as experimental lines, while 4 patients with a wild-type Neurexin-1 gene were observed as controls. A 4th line was also characterized, in which a Neurexin-1 deletion existed, but the donor showed no ASD phenotype. Cells were harvested from the patients and induced into pluripotent stem cells, also known as iPSCs, which then allowed for the differentiation of those cells into neurons. The use of iPSCs with the same genotypes as the donors allowed for the characterization of the Neurexin-1 knockout experimental lines, along with other possible contributing factors from their genetic background. 2 of the experimental lines contained a deletion of exons 1 through 3, while the third contained a premature stop codon, all of which result in a loss of function for the Neurexin-1 gene. To characterize our neurons, a microelectrode array was used. The MEA assay involves the culturing of neurons upon electrodes at the center of an MEA chip. Once attached, the electrical output of the neurons can then be measured to investigate the patterns of action potentials, or spikes, which occur across clusters of neurons. Many spikes in a short interval of time, also known as bursts, are indicative of robust communication between neurons. The MEA device is able to record the activity of a given culture and from this, measure when a burst occurs across the chip. It can then give information about metrics regarding the strength of the activity of the chip and the bursts themselves, such as average firing rate of action potentials, amplitudes of spikes, and burst duration. It can also show the path through which a signal travels, and from this devise the likely location of axons between neurons. So far, recordings up to the time point of day 63 have only been gathered for the 7880 line, which is a Neurexin knockout line, and C3141A, which is a wild-type Neurexin control. From this preliminary data, it can be shown that the Neurexin knockout line exhibits higher spike amplitudes across all time points. If confirmed, this could hint at a phenotype of higher voltage spikes within neurons with a loss of function of their neurexin gene. While less of a stark contrast, trends were also seen in which neurexin knockout lines had lower intervals between spikes and shorter durations of bursts. Further experiments with the additional experimental lines could help to solidify these differences and determine with more certainty if such differences in communication occurs between neurexin knockout neurons and their control counterparts. While the data presented only signifies the first batch of recordings, there were considerable differences between the amplitude, interspike interval, and burst durations of the 7880 line and the C31418 line. The increase in amplitude and decrease in burst duration and interspike interval could ultimately hint at shorter, more tightly clustered, and more powerful signaling from the knockout lines. Through the characterization of more batches of experimental lines, we can hopefully clarify the preliminary differences observed and possibly identify new areas in which the two different lines diverge. We hope to confirm the data gathered from MEA recordings with supporting evidence from patch clamp electrophysiology, which looks at the electrical charge within the membrane of singular cells. Finally, we continue to look for new ways to analyze the data collected from the microelectrode array by investigating more advanced methods of sorting through spikes to faithfully identify neurons and bursts occurring within cultures.